The overall goal of this procedure is to compare the concentrations of many proteins between samples by mass spectrometry using ReDi proteomics. This is accomplished by first digesting the protein from each sample to generate peptide mixtures. Next, the two samples are mixed and peptides in the mixed sample are fractionated and desalted. Next, the mixed sample is then analyzed by mass spectrometry. Finally, the peptides are identified by comparing the MS2 spectra to a target decoy database of all potential peptides in the samples, and the relative abundance of peptides between the samples are quantified. Ultimately, ReDi proteomics enables the quantification of the relative abundance of many proteins between complex samples.
The main advantages of reductive dimethylation over other methods for stable isotope labeling, such as SILAC, is that ReDi is a fast, inexpensive, and accurate method that does not require specific oxytrophies or a synthetic medium, meaning it can be applied to nearly any type of sample.
To begin, prepare 1 milligram of cellular protein in 500 microliters by using sonication or a French press to lyse the cells. To precipitate the proteins, add 1 volume of trichloroacetic acid to 4 volumes of protein and chill on ice for 10 minutes. Centrifuge at 12,000 Gs for 5 minutes at 4 degrees Celsius and remove the supernatant. Then use 1 milliliter of ice-cold acetone to resuspend the pellet before spinning a second time. After aspirating